Here, let us perform an activity to learn about the need of sunlight in the formation of starch. Click each tab to know more.
Take a plant having destarched leaves, that is, a plant which is placed in the dark for 3 days. Next, wrap the leaves of the plant with black paper on which a design is cut without removing the leaves from the plant. Now, fix the black paper on the leaf with the help of clips such that light should not enter the dark part. Now place this plant in sunlight for a few hours. Later, detach the leaf that is covered by black paper from the plant and test it for the presence of starch by pouring iodine solution on it. It is observed that only the portion of the leaf which is exposed to sunlight turned to blue-black and the remaining portion of the leaf which is under the black paper remained the same. From the activity, we learn that starch is formed only at the parts where sunlight falls. The parts of the leaves which turn into blue-black color by pouring iodine solution on it indicate the presence of starch in leaves. Thus, we can say that sunlight is necessary for the process of photosynthesis to form starch. Here, let us know about the presence of chlorophyll in leaves other than green color. Generally, we find leaves of plants with different colors other than green. Does these leaves also contain chlorophyll? The answer is yes. In these leaves, a large amount of red, brown, and other pigments mask the green color. The process of photosynthesis also takes place in these leaves. Also, you might have observed some green-colored patches in ponds or in other stagnant water bodies. These green-colored patches are generally called algae. Do you know why they are green in color? The presence of chlorophyll gives them the green color. Algae can also prepare their own food through the process of photosynthesis. Let us study about the synthesis of plant food other than carbohydrates. We have learned that plants synthesize carbohydrates through the process of photosynthesis. But do you know what carbohydrates are made up of? Well, the carbohydrates are made up of carbon, hydrogen, and oxygen. Carbohydrates are used to synthesize other components of food like fats and proteins. But proteins are nitrogenous compounds which contain nitrogen. How do plants obtain this nitrogen? The bacterium that is present in the soil converts gaseous nitrogen into a usable form and release it into the soil. These usable forms are absorbed by the plants along with water. You might have also observed farmers spraying or adding fertilizers to the soil which are rich in nitrogen. In this manner, plants fulfill their requirements of nitrogen along with other constituents. Plants can thus synthesize components of food other than carbohydrates like proteins and fats. Now, let us learn how to preserve the plants and its parts. Traditional designery art is a way of preserving the parts of the total plant like leaves, flowers, etc. Suppose the plants are not available in a particular location, then the physiologists collect those plants where they are available and make them in preservative form. This process is generally referred to as herbarium. The word herbar refers to a collection of preserved plant species. These preserved plants are studied by the physiologists and are also helpful for future studies. So far, we have learned that plants produce starch. Now, let us learn about other products that are produced by the plants. In the previous chapter, we have studied that our food contains starch, fats, and proteins. Similarly, all these substances are also present in the plants. First, plants produce sugar. Later, this sugar is converted to starch and to other compounds. But this conversion of sugar to starch involves many nutrients. The important nutrients include nitrogen, potassium, and phosphorus. Other nutrients are also required, but in minute quantities. These nutrients are termed as micronutrients. Plants absorb these nutrients from the soil. Studying of these nutrient elements by conducting experiments is very difficult. Now, we will learn about the other modes of nutrition in plants. Cuscuta is a thread-like plant which contains yellow structures twining around the stem, branches, and leaves of other trees. It does not have leaves and chlorophyll. It takes food from the plant on which it is climbing. Just like animals and humans, this type of plant species also depends on plants for food. This mode of nutrition is called heterotrophic nutrition. Thus, the plants that depend on other plants for food are called parasitic plants. These plants develop special roots called haustoria. Haustoria are root-like organs that penetrate towards the tissue of a host plant and absorb food materials from them, and sometimes they may even kill the host plant. Here, let us know the plants that grow on dead and decaying matter. We often find some umbrella-like structures growing on the ground after rains. These types of plants are called saprophytes. These saprophytes grow on dead or decaying matter. Similarly, you might have also observed some black and white spots on bread pieces, pickles, whenever they are kept in moist places. These black and white spots are also a type of plant that are developed due to the growth of fungi. These plants do not contain chlorophyll. Generally, they absorb the organic material from the decaying matter. Let us observe the fungi on bread mold by doing an activity.
Click each tab to know more.
To observe fungi on a bread mold. To perform this experiment, the following materials are used. They are spoiled bread, permanent slide, plucker, cover slip, and microscope. Take a slice of spoiled bread and a permanent slide. Now, place some rotten part of the spoiled bread on a permanent slide by using a plucker. Next, add a few drops of water on the slide and cover it with a cover slip. Now, place the slide under the microscope and observe it. Whitish thread-like growth with masses of black, grey and green fine dotted structures are observed. The tiny thread-colored structures that are found on the bread are generally called mould. This mould develops on the spoiled material and it has the capability of producing new mould. The common bread mould plant consists of fine thread-like projections called hyphae and thin stems having knob-like structures called sporangia. Sporangium in singular. Each sporangium contains hundreds of minute pores. When the sporangium bursts, the tiny spores are dispersed into the air. Let us learn about the special mode of obtaining nutrition. The plants that can prepare their own food and also obtain their nutrition from the insects are called insectivorous plants. These plants are also called carnivorous plants. The leaves of these plants are specially adapted to catch insects. As these plants are green in color, they can manufacture their own food. These plants generally grow in the places where there is nitrogen deficiency. They meet their nitrogen requirements from insects. Some of the examples of these plants include Drosera's, Utricularia, Venus flytrap, and Nepenthes cassiana. In some plants like Dal family, a type of bacteria grows on their root nodules. This bacteria acquires shelter in the roots of the plants and fixes nitrogen for the plants. This process of association is helpful to both bacteria and plants, and it is called symbiosis. The relation between algae and fungi starts when a type of fungus attacks the algal colony. This is an example of balanced parasitism. The algae and fungi live together in a symbiotic relation, forming colonies. These colonies are called lichens. The relation between algae and fungi starts when a type of fungus attacks the algal colony. This is an example of balanced parasitism. During the course of time, the algae survive and the fungus colony protects it from intense sunlight and conditions of drying up. The algae serve as the food provider for the fungus. In simple words, we can say that fungus gets food from algae and in return, it protects the algae from the intense sunlight and conditions of drying up. Let us study how nutrients are replenished in the soil. In some plants like dal family, a type of bacteria called Rhizobium grows on their root nodules. This bacterium acquires shelter in the roots of the plants and absorbs nitrogen from the atmosphere. It further converts nitrogen into ammonia and nitrate and helps the plant in their growth. Rhizobium cannot prepare its own food. It depends on plants for food and shelter. This type of relationship between bacteria present in roots and plants is called as symbiotic relationship. This association is of great importance to the farmers because they do not need to add nitrogen fertilizer to the soil. Types of leguminous plants include pulses like gram, peas, moong, beans, etc.
Follow-up work. Take up the following activities.
Get a sample of urea from the fertilizer shop or a farmer near your house or village. Sprinkle the urea near the plant whose leaves are turned to yellow color. Observe the changes in the leaves after sprinkling urea. Note all your observations in a notebook. Try to differentiate the plant before and after the sprinkling of urea. Collect a leaf. Take the peel from both sides of the leaf. Put the leaf under the microscope. Observe the number, size and shape of stomata. Note all your observations in a notebook. Attempt to draw the shapes of the stomata present in the leaf. Select any broad-leaved plotted plant. Take a cardboard and cut into desired pieces. Fix the cardboard pieces on the selected leaves. Put the plant under the sun for 7 days. Remove the cardboard and observe the changes in the leaves. Repeat the experiment on a number of leaves to get designer leaves. Note all the observations from the initial point of selecting the plant till you achieve the designer leaves. Visit a greenhouse if there is one near your place. Observe how they raise plants. Find out how they regulate the amount of light, water and carbon dioxide to grow the plants. Try growing a sweet potato just in water. Describe your experiment and observations. You have successfully completed the chapter Nutrition in Plants.